We've reached the third and final installment of Chapter 2, Chemistry. We've already talked about 2 of the 4 organic molecules. We've talked about carbohydrates and lipids. Here we're going to talk about proteins and DNA RNA. So proteins have a wide range of functions. They're made up of amino acids, and they perform lots of processes inside our cells. We've got them for muscle contraction. They are pumps on the outside of our cells, and enzymes. Proteins are made up of amino acids. Amino acids are all very similar in structure. They've got an amino group, a carboxylic acid group, hydrogen, and then an R group. And that's where they're different. Different amino acids have different R groups. And those different R groups have different properties. Some are going to dissolve well in water. Some are not going to dissolve well at all. Some are going to be nonpolar. And the specific order of amino acids determines what a protein looks like. So you've got these different levels of organization. First, the order of amino acids is just the primary structure. And what happens from there is they either form the secondary structure of an alpha helix or a beta sheet. And from there, those come together for the tertiary. And then quaternary is where you have multiple tertiary structures together, and that's a whole protein. Enzymes are an example of proteins that are a catalyst. So remember, those make reactions go faster by lowering the activation energy. They are highly specific. For each reaction, you need a different enzyme. Each enzyme only works for one reaction. They are extremely efficient, increasing the rate of reaction thousands of times to millions of times faster, and are subject to cellular controls. So that's big. We get to control what happens in our bodies and in the cells based on what enzymes are present. We can turn an enzyme on, or we can turn it off to make the reaction occur or not occur. So here's what it looks like. An enzyme has what's called an active site. This is where the reaction takes place. It binds in the reactants, does the reaction, and releases the products. And you can see at the end, the enzyme is the same as before it started, so it's ready to go again. The enzyme doesn't get used up during the reaction. It's ready to go. It can catalyze multiple reactions in a row. Last, we've got DNA and RNA. So these are our genetic code. In our cells, our DNA sits in the nucleus, and every cell has the same DNA. These long strands of genetic information that code for proteins. So what happens is we take DNA, we turn it into RNA, that leaves the cell's nucleus and becomes proteins. That tells us the order of amino acids. The structure is on the outside. You've got a phosphate group attached to a sugar. And attached to that sugar is a nitrogenous base. And there's only 4 different nitrogenous bases. In DNA, you've got adenine, guanine, thymine, and cytosine. So what happens is, on one side, you'll have one nitrogenous base and it pairs up with another on the second strand of DNA. So T-thymine always attaches to A-adenine. G always goes with C. So when we want to read the DNA to make RNA, we have enzymes come in, pull this apart, and then we read one side to make RNA. So RNA is single-stranded. DNA has these 2 strands that form the double helix. RNA only has 1. And another difference is that RNA doesn't have thymine. Instead, it has U, uracil. So anywhere there's a T, RNA would have an A. But anywhere there's an A, RNA would have a U. So you can see the difference in nitrogenous bases. DNA has T, RNA has U. There is a difference in the sugars. DNA is deoxyribose, whereas RNA is ribose. Number of strands, DNA is double helix. RNA only has 1 strand. DNA in each cell is self-replicating, so we read it to make new DNA, whereas RNA is made from DNA as a blueprint. Then down here, we'll talk about mitochondrial DNA more in Chapter 3, and then the different types of RNA. It's really neat that there are 3 different types of RNA with different functions, but they all work to code for proteins, and we'll look at how they do that next chapter as well. The last thing I want to have you guys see is adenosine triphosphate, ATP. This is the universal energy source for all organisms. So we've got there is an adenine, that's the A nitrogenous base from DNA. You've got a ribose sugar, and then you've got 3 phosphate groups. Notice ATP triphosphate, that's 3 phosphate groups. 1, 2, 3. So these bonds here, the wavy ones, are really high energy. So what happens whenever we want to pump a pump or move something in the cell or make a bond is we break one of these bonds. It releases energy, and we use that energy. So after you break one bond, you've got adenosine diphosphate, ADP. And if you break another bond, you would have adenosine monophosphate, just the one phosphate left. Okay, and that's all I have for chemistry.